Last time, we laid the dinosaurs to rest. In this final lesson, we'll take a look at whether we could somehow bring dinosaurs back to life. Let's begin our story back in 1990 with the publication of Michael Crichton's novel Jurassic Park. The book, and later the film, were built around the notion that a wealthy entrepreneur had discovered a way to tap into damaged dinosaur DNA they found preserved in ancient mosquitoes trapped in amber. With the tools of genetic engineering, they spliced the dinosaur DNA together with that of living reptiles, birds, or amphibians to fill in the gaps of the genomes that were lost to time. As a result, they bring back a diversity of dinosaurs from the dead and create a dinosaur theme park on an island off the coast of Costa Rica. The problem? The dinosaurs eventually run wild, wreaking havoc for the humans who get caught up in the mix. Luckily, among the expert scientists they bring in to evaluate the park before it opens are a vertebrate paleontologist and a paleobotanist who use what they know about the lives and worlds of ancient dinosaurs to save the day. It's a fantastic story built upon lots of fragments of science, and it captured the imaginations of the public and dinosaur scientists alike. I can still remember the first time that I saw Jurassic Park in a theater in Bozeman, Montana. I was just finishing up my first year of college, and my advisor and mentor, Dr. Jack Horner, was the science consultant for the movie franchise. Our whole paleo team of grad students, fossil preparators, and collections managers got to attend a showing of the movie in Montana, where the opening scenes of the book and film take place. It was quite a night. Since the 1990s, paleontologists have been hard at work to try to do some of what Michael Crichton's creative mind inspired. We are on the hunt for molecular data that might tell us something about how dinosaurs lived. And yes, we are working to understand just how dinosaur genetics shifted to result in the advent of birds from their theropod ancestors. Jack Horner continues to be at the forefront of this area of research, tapping into the science of evolutionary developmental biology to try and rewind the history of evolution using the embryos of modern birds. But he's not alone. In molecular biology labs all over the world, scientists are examining the record of extinct DNA preserved in modern bird genomes to figure out the ways in which evolution changed running, sharp-toothed dinosaurs into toothless, flying birds. This kind of research may have you wondering, could we someday resurrect dinosaurs? And as Michael Crichton asked in Jurassic Park, even if we could, should we bring dinosaurs back from the dead? We'll divide this lesson up into two big parts. In part one, we'll cover the possibility that fossilized bones from non-avian dinosaurs could yield soft stuff, including preserved proteins or DNA. In part two, we'll come from the other direction and explore how molecular biologists are mining the DNA of living birds to identify how they evolved from dinosaurs. If you've been interested in dinosaurs any time in the last 20 years or so, there's a good chance that you've heard news reports touting the discovery of dinosaur soft tissues, including things like preserved blood vessels, little bits of organic bone material, bone-building cells, and even possible red blood cells. We've been on the hunt for the preserved soft parts of dinosaurs since at least 2003, when the first hints that blood vessels and cells were thought to be recovered in demineralized bone fragments from a 68 million-year-old Tyrannosaurus rex. Chemical analysis of these unexpected squishy parts revealed the presence of collagen, the protein that helps build bones at the microscopic level, and imparts a little elasticity to the skeleton. This finding blew paleontologists and geologists' minds and stirred a vigorous debate in the field. How in the world could proteins survive the vagaries of the fossil record, sticking around for tens of millions of years? Since the first paper appeared in 2005, follow-up studies have come out at a rate of about one every couple of years, each asking a different question. In the first follow-up paper, researchers ran an evolutionary analysis that included the protein sequence data, basically the pattern of amino acids that fold and twist to make the specific collagen molecule, and compared it to the protein sequences of living chickens. For good measure, they also sampled the preserved collagen sequences of a fossilized mastodon and living mammals. The results supported the hypothesis that the collagen recovered really belonged to a T-Rex and the mastodon, and it correctly placed the T-Rex within Archosauria, alongside its alligator, ostrich, and chicken relatives, while grouping the mastodon with living elephants. The next paper took a closer look at the tiny, soft, transparent structures recovered from the T-Rex and from a hadrosaur, Brachylophosaurus. These structures resembled modern osteocytes, which are the cells that maintain bone in living animals. To test the hypothesis that these cell-like structures were original, the team borrowed techniques from modern immunology and chemistry. They were able to confirm the presence of small amounts of DNA and were able to rule out that the DNA they found belonged to bacteria. So, if there are potentially DNA, cells, and proteins preserved in these very ancient fossil bones, how could they have made it through the long and arduous process of fossilization? It turns out it might be all about chemistry, and those proteins may not be quite what they seem. The traditional story of fossilization is that within a few million years, or maybe even less, all proteins in animals' soft parts should be completely degraded. A team from Yale University studied the chemistry of bones, teeth, and eggshells from animals ranging from the late Triassic to the modern era. Of course, all the modern specimens contained soft tissue, but so did many of the fossil samples. The scientists used a method called Raman microspectroscopy, which doesn't require destructive analysis, but instead bounces a laser off fossils to reveal their molecular makeup. They found that these strange, soft structures were not made up of original dinosaur proteins. Instead, the structures were made of new, chemically transformed compounds that replaced the original protein molecules while preserving the overall shapes of the original soft parts. The shapes remained because these newly formed chemical compounds resist degradation and dissolution. They stick around. Bottom line, though actual dinosaur DNA might be hard to come by, other detailed data on soft parts are available in the fossil record of dinosaurs. That is exciting news for paleontologists. So if we can't readily access ancient dinosaur DNA, what might the DNA of living dinosaurs have to say about the unfolding evolutionary story that led to all the adaptations exhibited by modern birds? That's part 2 of our story of resurrection. You may have heard a rumor floating around that scientists in labs were doing some crazy experiments to transform a chicken into a new animal, one that more closely approximates its dinosaur ancestor. Some people call this creation Chickenosaurus. The idea is built upon our rapidly expanding understanding of the genome, the complete set of genetic material present in an organism, especially the genetic wilderness of the in-between regions of protein-coding genes. Some of these stretches of DNA include what are sometimes called pseudogenes, but I like to think of them as fossil genes. These are the ancient genes carried along through evolutionary history by being passed on from ancestors to descendants. Over time, these fossil genes are modified by mutations that have changed their function, sometimes by so much that these once useful genes are no longer needed. They continue to come along for the ride in modern creatures, though, because it is too difficult for evolution to throw them away. That's good for us because it gives us a window into the past, if you know how to look for it. The field of study called evolutionary developmental biology, or EVO-DEVO for short, does just this by combining the fields of evolution, genetics, developmental biology, and paleontology to investigate how changes in deep time may have occurred on a molecular level. This research area is super cool to me. I've always been the kind of paleontologist that works on old dead bones. I've never spent much time thinking about the molecular record of evolution preserved in DNA because, well, it's not preserved in the dinosaur fossil record. Through a series of experiments back in the 1970s, people working in Evo Devo started looking at model organisms, especially fruit flies, to try and understand how DNA actually works in building new structures. Sometimes a mutation would occur that might result in a fruit fly hatching with legs where its antenna should be, or an eye where its antenna should be. You can think of these weirdo mutants as evolution's monsters. These discoveries led to the detailed study of gene expression, how the genome directs the building of animal bodies. One of the key discoveries of molecular biology since the early 2000s is the realization that even though all animal life looks wildly different, we are genetically quite similar. We have major genetic programs for organizing our basic body plans and building the amazing anatomical structures that are conserved across many different animal groups. You might think, there's no way that we share most of our genetic code with penguins or dinosaurs, but the reality is, we do. By expressing some genes but not others, very different animals can come to evolve over vast amounts of time. This means that the genome is like a fossil record of molecular evolution that can be mined for the discovery of fossil genes, kind of like when we excavate dinosaur bones from the rock record. By exploring this genetic fossil record preserved in living animals during embryonic development, Evo Devo scientists are able to answer specific questions about the genetic underpinnings of major evolutionary transitions. Their goal is centered on trying to understand how small genetic changes can result in the development of innovative new structures and morphologies. So long as these genetic mutations aren't fatal, they can be tried by natural selection and eventually lead to the evolution of new, fantastic groups of life on Earth. Let's take a look at a few case studies in which researchers have succeeded in making the limbs, face, and tail of unhatched chicks get a little bit closer to the shapes we see in dinosaur fossils. The trick is a little genetic tinkering with the molecular pathways that build these anatomical structures. Remember our discussion of the semilunate carpal, that extra special half-moon-shaped wrist bone arising in theropods that are closely related to modern birds? It turns out that later birds went even wilder with their wrist remodeling, shifting from as many as 9 individual wrist bones in early dinosaurs to only 4 bones in living birds. One of the challenges has been telling which wrist bone is which. In other words, which dinosaur wrist bones are the ancestral bones for the simplified wrists of birds? Embryological study has helped solve this mystery. In some cases, a single bird wrist bone results from the embryonic fusion of one or more dinosaur elements. In other cases, birds have reacquired bones that were lost in their dinosaur precursors. With only the fossil record at hand, we might misidentify bird wrist bones. With only the developmental data, we might underappreciate the evolutionary history of these innovative features. It's the combination of data from evolution and development that makes this approach so powerful. You might not realize this, but embryonic alligator skulls look a lot like the skulls of adult chickens. Fossilized baby dinosaur skulls show a similar pattern. Their little skulls look a lot like the skulls of adult birds. This interesting morphological similarity prompted Arcot Asbenoff, then a biologist at Harvard, to propose an interesting hypothesis. Could dinosaurs have shortened their skull development early on in life to result in a more bird-like skull anatomy? This idea is about what is called heterochrony, essentially how the timing or rate of development can result in dramatically different morphologies. We think these small developmental shifts can help drive evolutionary history in many major vertebrate groups. Arcot and his team collected tons of data on ancient embryonic dinosaurs and early birds, tracking the morphological changes across the dinosaur-bird transition. He identified a few different examples of growth truncation, called pedomorphosis, essentially the retention of juvenile style characteristics into adulthood. We see this as it relates to the shortening of the snout and the enlargement of the part of the brain associated with vision. He also identified an example of an elongated period of development that results in morphological change. This lengthening of developmental time is called paramorphosis. The bones that build the beaks of birds represent paramorphosis, since their development is stretched out in birds. These findings indicate that over evolutionary timescales, modern birds become more and more baby-like in their appearance. Essentially, a modern bird looks like a baby dinosaur, just one that is able to reproduce. Next, Arcott's team turned their attention to the genetic mechanisms that build bird beaks. Those toothless tools are great for all kinds of things: sipping sap or nectar, cracking seeds, drilling trees, sieving water, pruning, and nest building. You name it, and bird beaks can probably do it. Beaks are sort of like having fingers on the face. It turns out that just a few minor genetic changes can morph a beaked chicken embryo into a dinosaur-style beakless face. Here's how they did it. The beaks of birds are made of two fused bones at the front of the face, the premaxillae. These are the same bones that you have located where your front teeth are in the upper jaw. Like dinosaurs and alligators, our own premaxillae are unfused. They just sit side by side in the jaw. To figure out how the fusion in the bird premaxillae happens genetically, Arcott's team mapped out the activity of two different genes that are expressed in the separate premaxillae of most living animals. Reptiles and mammals have two patches of gene expression, one for each side of the nose. Birds only have one for in front of the nose. Since dinosaurs also have separated premaxillae, we can assume an alligator pattern of gene expression in dinosaurs. The researchers then turned off the expression of the genes in chick embryos, but only in the middle part of the face. The result? They created a chick embryo with a skull that resembled that of a dinosaur or an alligator rather than a bird. Once again, we see how a small change in genetic programming can have big impacts on the morphologies of final anatomy. One of the most recognizable differences between the skeletons of birds and most dinosaurs is the short little tail of birds known as the pygostyle. The transition to this new tail anatomy was probably a pretty important event in bird evolution, and it seems to have occurred over a relatively short evolutionary interval. These short little tails are critical components of powered flight strategies of birds. They are also connected to feathers employed in mating displays and are used to communicate, wiggling and flashing to warn other birds when danger is nearby. The pygostyle is present in the entire diversity of living birds. We have a great fossil record that documents the anatomical transition from the long and flexible dinosaur tail to this newer, shorter form in birds. But how did this transition occur developmentally? The external phenotypic changes that we can see in the anatomy of birds relative to their dinosaur precursors result from changes that occurred during embryonic development. This means that we can investigate an array of potential genetic mutations in embryonic model organisms that may have been responsible for the evolution of stubby tails. Research is focused on two kinds of model organisms, mice and chicks. Though the research can't pinpoint exactly what mutations occurred early in bird evolution, or even which dinosaur was the true ancestor that exhibited the original mutation, we have learned enough to get closer to understanding how chickens might have lost their long tails. Genetic and fossil evidence indicate that it's possible a single mutation could have occurred in a feathered Maniraptoran dinosaur. A current working hypothesis is that the genetic change, or changes, occurred in genes that are involved in lengthening the vertebral column. All of this amazing genetic work is providing incredible insights into the toolkit that builds bodies and how small genetic changes can have a big impact. Though we may never really be able to resurrect a dinosaur from a living bird embryo, it's still pretty amazing to see just how far we've come and how much we've learned about all the molecular things that made dinosaurs dinosaurs. Besides, would you really want to live in a world where a T-Rex, Carnotaurus, or Velociraptor could hunt you for food? Over these 24 lessons exploring the age of dinosaurs, I hope that you've gained a deep, new appreciation for these fantastic beasts. We've investigated the origins of dinosaurs and delved into how paleontologists divvy them up based on key anatomical differences. We've also dug into the details of dinosaur anatomy and paleobiology, taking hints from their living descendants to dissolve some long-standing myths. And along the way, we've painted a picture of a dramatic, diverse, and interesting group of animals. Once, one of my college students asked me, given all you know about the story of dinosaurs, their survival through major mass extinctions, and even their continued dominance of the sky in our living world, are they the true rulers of life on Earth? It's a great question. The Dinosauria certainly puts us humans into context and drives the point home about how lucky all of us are to have relatives that squeaked through that most recent mass extinction event 66 million years ago. In the time since, humans have evolved the intellect that allows us to dive into the past to exhume and understand these Mesozoic monoliths. Though we are living in the age of humans, I hope you've come to see just how remarkable the age of dinosaurs actually was. Thanks for joining me on this journey to rediscover it.